Okay, good afternoon, everything. Let's start our workshop plant-based metabolomics. Uh, Dear colleagues and students, it's my pleasure to start our workshop, and we change a little our program because we have Professor Castro Gamboa from Araraquara. He's had a fever and test a positive for COVID. Then came he say sorry about it. And he had more one professor, Moacir was Forim, Moacir go in the morning for me, he had a problem with his mother, and he needed to travel to Paraná, and I understood Moacir because for me, family is very, very important. Okay, first of all, I would like to ask our professor. I forgot Tiffy, head of our department, Professor Clever, to open our workshop. Please, Clever.
Okay, good afternoon for everybody. It's my great pleasure to welcome all of you. On behalf of the Department of Chemistry, I'd like to say a special thank you to Professor Fatima for inviting us. I always say that natural products chemistry is very important issue for us. It's a very traditional area of our department, and we are very proud to have all of you here. I'd like to say my special thank to Professor Choi for giving us this very long week of courses. It's a very pleasure for us to have it here. And I wish all of you take this time for learning more about natural products and enjoy your time here in our department. Thank you.
Oh, thank you, Clever. Okay, then let's pleasure now to introduce the first speaker, Professor Hae Choi. He received his PhD degree from College of Pharmacy in Seoul National University in Seoul, Korea in 2000. He came into division of pharmacognosy section, Metabolomics, Institute of Biology, Leiden University, as a postdoc in 2002. He became a researcher in the section Metabolomics at the Institute of Biology, Leiden University, the same university, from 2003 to 2008. In 2009, he became assistant professor and associate professor at 2014, and he is a scientific director of metabolomic facilities at Leiden University since 2013. And exactly why he was here to give a course during this week in metabolomics. And the course was excellent. The students could learn a lot about metabolomics, and everybody could learn that metabolomics is a tool. You can use metabolomics to help chemosystematic, to help your work if you intend to analyze the interaction insect and plant, and all the other analysis and the other interaction. Only more one, I think it was very, very important for the students. How they could learn that they needed to think about their work, about his work. They needed to think what they needed to do, what if the objective is exactly the correct objective, if what they planning to follow in the analysis, is it correct, or what is the question, and how can we answer the question. I think it was very, very important for the student. And the same again, Choi about the course, it was excellent. I'm sure the students could learn a lot. Thank you, Clever. You can start, please.
In case, can I have more minutes?
Yeah, yeah.
Thank you very much for introduction. In this week, I was introduced 2 times. Probably you are quite boring, or you can remember. Getting old, sometimes, it's not only getting more age, sometimes your mental passion cannot, physical passion cannot follow the mental passion. Today I was a bit got a cold, so please forgive me if my voice is some changing. So this week I gave a course almost 25 hours, right? 25 hours from starting from biochemistry. Here, just this morning we finished the statistic, how to analyze the data. And as I mentioned also, Professor Van der Laan, Professor Fatima mentioned, using these metabolomics and excellent tools, how can we apply it to natural product research? This morning, Professor Van der Laan shows one of the revisiting natural product with a tool of metabolomics to see chemical treasury as a natural product. Of course, here is the organic synthesis. Professor is here, but still natural products, 50% of new drugs are coming from, I don't have to mention, 70, including the semi-synthesis, more than 70% are coming from natural product. But real situation is when you talk with big pharma, they are not very much interested in natural product. Do you know why? Presentation for research, lecture should be different. Today I still is keeping my way to teach the students. I should not ask you, but maybe if it's a flexible way, I can keep this way. Because it's a complex. This is simple. Because it's a complex. Simply is a one word. Is the difficulty to use natural product because it's a complex. Then probably we need some other aspect. Depending on the aspect, maybe we can see another advantage. That to take this one, probably we can use metabolomics. Then natural product, everyone think is the treasure or the storage of all the natural the bioactive compounds. As I mentioned, 250,000 chemicals reported already in a single plant material. 30,000 chemicals are expected even in plant material, but our real situation, we cannot make them interested so much with the natural product because of the complexity. Potential is higher. A real outcome is sometimes we have a bottleneck. In that case, using metabolomics, we can approach in other aspects. My case is a little bit different from others. I want study natural product as a new system. One example is today I took antibiotics because I got I'm supposed to have an infection. But when you look at the plant material, plants in the garden, plants never take antibiotics. And also when you look at very detail about the antibiotics in the market, 99% antibiotics are from microorganisms. Plants never produce proper single antibiotic. Over a million years, how it's possible plants can survive with the numerous pathogens without drugstore containing antibiotics? Then, simple question is, probably they have a different system. This system, so-called, it can be the new target of natural product that metabolomics could help for you to take that system. Then nature is a source of bioactive compounds. No doubt, it was very well explained this morning from the lab, but also the treasury of valuable system. This system, what kind of system it is? So every 5 years in Netherlands, I have to change the topic. Because if I keep same topic, then everyone is blaming me. You are doing every time same topic, so I have to change it. Last 5 years, I have focused on this one. I'm working for the natural product since I got a master's degree in Seoul National University under the pharmacy. Then maybe is that anyway now natural product has a potential, but there's just some we are losing the interest from others, attention others. Then we can divide it for category. Material, probably if most working, my case is not your case, I'm working with the medicinal plants. Maybe medicinal plants is not enough. Maybe we can change to other organisms. That is another possibility. Either microorganism, or you can go to the deep sea, the marine organism, or who knows, next year I'm going to the Mars or Moon to take another new sample. That's if as long as you want new compounds, you need it. Second one is technique. Of course, I'm not engineer, as I mentioned, I'm not engineer. Probably someone mentioned, now we can see only hundreds of compounds, or 30,000 compounds from plant material. But next year, with the new technique, who knows, we have 3 million compounds in plant material. Then we can increase the number of compounds as a potential of new drug. The other one is synergism. That is important one. Synergism, everyone believes, even my mother believes, if you take two, then you have a new activity, but no one can prove. That is an important one. Yeah, right? Cachaça with a lemon, very nice synergism. But cachaça itself is not very nice, yeah, because I got a problem last Wednesday. This Wednesday, I took only cachaça with a straight. Next day, as you see, I had a lot of sweat. I had to drink with the lemon or lime cases. That's the synergism. The other one is interaction. Interaction means everyone believe I extracted the medicinal plant or I'm human. But actually, you look human, but inside of your body, you have a lot of microorganisms. You are bugs or humans or mixture cases, so interaction cases. Those four topics I tackled with the metabolomics. Today, of course, we have a very limited time, but I can show some simple example. It's not new. Sometimes I published it 10 years ago, 5 years ago. It's not very new, but I want to give some kind of a new approach how to do it. So new revisit that is new of the microorganism and marine organism. So traditionally medicinal plant, but most of the new chemicals are from microorganisms nowadays. And marine natural product can be the new source. And specific parts of organs can be possible, specific parts. Even you are doing plant material. I will show you later. Now, latex or exudate, cells can be target of the compound, not like a whole plant material. Marine organism cases, completely is a very very interesting material. Why? Because they are living in different media. For example, now we have a fatty acid. Yeah, we have a terrestrial organism, fatty acid, but some organism living in seawater, particularly seawater, the fatty acids, many conjugated bromide, iodide, or those things, because media is a completely different one. Maybe those kind of a change can be the source of new compounds. So marine organism case is a unique chemical diversity. When you look at the marine organism, first is chemical diversity. But you cannot use properly the database. Why? Because chemical diversity is a completely different terrestrial plant material. And second one is mostly, maybe when you tested the plants outside of your garden, 1 out of 1,000 active against your bioactivity test. Marine organism cases, if you selected 10, more or less 50% is active. It's strange. Yeah? So, but bad news is taxonomically, it's very difficult. Taxonomically, actually, taxonomy, it doesn't matter. Someone said, oh, if I want to use the bioactive compounds from plants or others, taxonomy is very, it's not my concern, but actually it's not true. For the reproducibility, to get more amount of the compound, you need a taxonomy case. But the problem situation is, for example, spongy cases, when you collect the spongy, real situation, only genus name is specified, not species name, because they are living together, they are not alone cases. So taxonomy genetically also is impossible. Then difficulty to grow, of course, it's grow. Of course, San Diego, Scripps Institute, they are using robot or some marine, submarine cases. But when you not like a medicinal plant, you cannot harvest like by hand. You need a special instrument. So that's why you cannot increase the amount of the material. So when you do drug development, usually at the end you need 1 kilogram of the compounds, 1 kilogram of the compounds. But 1 kilo of the compounds to get the marine organism is impossible. So that's why when someone is doing marine organism, they are doing always is chemical synthesis at the end because they cannot get the compounds. And not yet marine and aquaculture, because some organism can be cultured, like a marine culture in the lab or aquaculture in the ocean with some fence, but few of them. Someone ratio is very low. Even you grow the marine organism, metabolize is completely different. And more interaction with the environment and organism, which means metabolites are all varied. So that's why we studied the first experiment is the marine sponge. We used this one giant barrel sponge. The most studied marine organism as a model sponge, we collected this one. I didn't. I sent some people to collect it. And a wide range of metabolites, but a great variation by location. So to define the metabolites, I want to know marine organism. First one is age. Yeah, depending on the age. Second one is same marine organism you can collect from Caribbean or Indian Ocean or Pacific Ocean. Are they different or not? And third one is sometimes it's depending on the deeps. But usually terrestrial plant material altitude is not really concerned. Always they are using in the field. But marine organisms, depending on the depth, pressure is completely different, and the light intensity are different. So that's why it's like this one. First, as a basic experiment, we did this. Then now we did it giant barrel sponge here, Curacao, Martinique, Taiwan, Tanzania. They are very, 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 very different. Now is the issue. Then how to get the compounds. Then now it's the first step to use not targeted analysis to use marine organisms. We have to get more information with the help of metabolomic cases. Then discriminating metabolites looks like this one, bromide, as you see. As I told you in the beginning, on the first day, the acetylene compounds in nature is not very common because it's oxidative. If any, it's a chrysanthemum volatile cases. But here in the lipid layer in marine organism, bromide one is there are a lot of the compounds are there. Also alkaloids there. But this one is not fixed within the range. They are always varied depending on the location, depending on the age, depending on the depth cases. So now we have to, of course, it takes some more time to use the real marine organism as an extract itself. Then this kind of metabolomic approach we need. So still, what difficult is in identification. Now, many people are using, Van der Laan mentioned the molecular networking with the GMPs, or I mentioned Dictionary of Natural Product and Animal Analysis. When you identify the compounds, always a bottleneck is how to identify the compounds because most of the compounds is not listed in your database. Now is the marine natural product is listed, it's getting increased, but still is very different. And also keep in mind, this one is sometimes when you are using the mass spectrum, the identification, many cases we are based on adaptive ion. Adaptive ion is like M plus H, M plus Na, M plus K. So based on ratio, we can collect it, but sometimes marine organism, the adaptive ion is different. And sample preparation, preservation, desorting, and sample preparation, as I mentioned, when you collect the plant material to the metabolomic, in the middle of ocean, you cannot bring your refrigerator, you cannot bring your liquid nitrogen, you cannot bring those kind of things. Many metabolites, small amount, they are changing. So that is a limitation. And another one is material method is, of course, marine microorganism we are using. But microorganism cases, if you were asked, how many metabolites do you expect in marine organism? As I said, maybe it's the same number as plants, but it varies a lot. Microorganism is their change, their potential is completely changed by environment. Sometimes with a very the happy situation, they produce very minimum metabolite. They don't have to produce it. But when they are together with the pathogen, metabolites are changing. Also, when they are with a very special medium, then they are changing. So that's why we did it together with some marine organisms like a fungus and bacteria, we grow together. Then bacteria case, fungus is pathogen. Then metabolites are increased. Toxic metabolites. Toxin means we can translate to the bioactive metabolites. Here, this one is one of the example. I think it's 2015, together with the Microsoft. Here, Streptomyces bacteria. Yeah? Streptomyces bacteria and Aspergillus. Aspergillus is the fungi cases. When you grow together, then you have new compounds, new metabolites are coming up. The problem is, now is good situation. You can use co-culture cases. What's the problem? To study metabolism? As I mentioned, our technique always we can have very limited number of metabolites. Out of 30,000 metabolites in plants or microorganisms, we can detect only few dozens, or maximum is 100 metabolites. Then this one is unknown metabolites. Always we are identified using biosynthetic pathway. Because already many people knows biosynthetic pathway from the CAC database. Then you find some metabolite. The other one is you can fill up with a known database. But the problem is here. Now, when they are living together, biosynthetic pathway is completely changed. As I mentioned, one example, to synthesize by plants, the terpenoid, many people grow together and the biosynthetic pathway is not correct anymore. That was the limitation. And to prove this one, we fed the one precursor, which is not in plant, which is not in bacteria or fungus cases, plant metabolites, we feed. Then here, new compounds coming out, not from bacteria, not from fungus, when they grow together. That shows potential. Maybe with the feeding experiment, co-culture can increase the number of metabolites or complexity or diversity of chemicals. Another one is, I told you in the beginning, the always bottleneck, also the barrier to use natural products as for a drug development is a complexity. It's not number of the compounds, dynamic range. Some metabolites, gram per kilogram, some metabolites, picomole, per kilogram cases. Then when you use this one, of course, they said we do some screening test. You find the metabolite, you would like to isolate the compounds, right? Then you can isolate the compound. Many people fail because it's a too low amount of the compounds. Although you can find sometimes new compound, new bioactivity, but then we can change some aspect. If you are working with a plant microbial interaction compound from plant material,
why we have to use plant itself? Maybe plants excreted some latex exudate. This latex probably is kind of a first battlefield between plants and microorganisms. Then plants excreted. My hypothesis, or your hypothesis, probably in the latex, very few number of metabolites, it's naturally filtered metabolites. Those filtered, those released or excreted metabolites, probably bioactive because they want to control these latex cases. And also important one is when you develop a new drug, it's not only target metabolites, when you target metabolites, but also drug formulation is very important. Like many kinds of bioactive compounds, is not soluble water or degraded, unstable cases. But when the plant material is treated, this one is not biofluid anymore under the sunlight or dryness. Probably they are unstable. But our nature, probably they might have a natural formulation reagent in this latex. So not only target filter the metabolite, few number of metabolites, but also we can learn from the plant material, maybe natural formulation can be included in the latex. That's our starting point. Then we need the metabolomic, the profiling basis. So this one is, it's like our hypothetical approach. Active components, probably those kind of metabolites included on protection from the UV light, also protection from the dryness. Because if it's only water under the sunlight, it's dry completely. This is we did it. Then I collected latex cases. I collected the latex, but completely different species. Many latex producing plant, Euphorbia plants or Aponicea plant material. I collected a different one. When you compare those different species, metabolic profiling, completely different one. And now I have this one, plant cases in animal cases, more or less this triterpenoid part here. Now, latex, you can draw conclusion. Latex cases compared to main plant, host plants compared to, they have a very high amount of terpenoids in general. Why? Then we can answer, we can have a hypothesis later. Or at least this kind of metabolomy can raise you new question. Why latex contains more terpenoid? It's a starting point. Yeah. And now we need a TLC. I think you like the TLC. It's a simple and also it looks like a beautiful. Everyone likes it. So now it's latex and leaves. Yeah? Compared to this one, in terpenoidal region, they have quite a lot. Yeah. And another one is I tested this one. I omitted one slide. When you tested the latex extract and host plant extract against an insect, always latex shows very high activity. Very nice, because we expected. Yeah, plants produce this one to protect themselves from insect or bacterial cases. Now, situation is here. Is one of them is Euphorbia myristina, and this one is Apocyna alstonia scolaris from Apocyna plant material. It looks like a pure compound, right? This looks, if you have a pure compound, this one is only 3 compound mixture. This one is only 1 compound. It's strange because I just take the latex, I added the water, I measured the animal step. Now, first, good news is my hypothesis can be proven. So in the latex, very few number of metabolites. But bad news is this kind of metabolites, although this one is a new compound, it's similar, it's never been in the nature, but this compound, it doesn't look like a very active, right? It's not very active. Then we have to answer to this result. What kind of answer do you have? Plant, in the beginning, as I mentioned, plant does not have to produce antibiotic. Why? Because they have a best friend, symbiotic microorganism or some endophyte cases. Then in this case, We test not this one, in this case, this metabolite is very good to break down biofilm. So now, story is, it should be more further proven, story is plant does not produce direct, very targeted antibiotic, but their endophytes or symbiotic bacteria produce antibiotic and they are helping. Probably this one is maybe signal molecule or to help the antibiotic cases. So then I mentioned triterpenoids. Terpenoids is very high in the latex. When we tested this latex, first peptide or protein is very high latex. Then they make a barrier. It's like our bandage. Then they protect it physically, bacteria or fungi, or this one. And second one, triterpenoid cases. Now I'm very sick today. Yeah. Then what should I do? Should I go to the emergency cases, 911? No, Tylenol. That is the best one because first, very weak one is very general one. I took it. This kind of a triterpenoid, interestingly, they don't have a very strong activity. My suggestion is, but this one is a very broad, like a Tylenol or ibuprofen cases. After that, why they then use this one? The peptides or like polymer cases, isopropyl polymer cases. First, physically protected, and then when they pass through this physical protected one, then Tylenol, like a terpenoid, retarded the infection, then the plants have a time to produce their compounds or symbiotic agents. So this is a time basis. We can learn from the nature basis. And now this one is a new material. Although it's a new material, everyone is Fatima or Vandelam mentioned the latex cases, but still it's with a new metabolomic tool, we can approach it for new aspect. Second one is technique. Technique it's not my concern. Every day, new technique of the mass is coming out. Yeah? Nowadays, it's not only adaptive ion, you need the ion mobility data or accuracy, whatever. But I wouldn't say I'm involved in the development of new technique, but at least our case, application on this, we have to understand where they are going. Then where they are going means what's the current issue in each technique. Those two techniques in the metabolomic, NMR and LCMS, that's the one. So their goal or issues are completely different. So here, first one is, you understand, all the students? Yeah. If you want to see the moon, you need a telescope. If you want to see microorganisms, you need a microscopy. But current situation, like a microscopy, LCMS, you can see details, but we cannot cover this. NMR, very nice to watch moon or star, but you cannot see microorganisms on the moon. One day we'll have this kind of one lens to cover both macro and micro game, but still we have a problem. So that's why even I said metabolomics is a new tool, but technique limitation is present because of the limitation of the techniques. Then this case is mass cases. Mass, you think your mass is the perfect? No, it's not perfect. Maybe 2 years later, some years later, maybe next month, new model is coming out. What is going to do mass technique? They always want to do accuracy and resolution. Why? Because the first one is mass, nominal mass, 300. If you search for the compounds, because you have to go to the approach the search, then it's a problem because then it's hundreds of compounds are coming out. So that's why they want to get more accurate data, precision data, so-called high resolution mass spectrometry. 4 decimal, 5 decimal, 6 decimal. Second one is fragmentation. Now fragmentation for several decades we ignored because we developed from EI spectrum, ESI spectrum, we suppressed the fragmentation ion under the name of ESI, APCI, or multi cases. But the problem is at the end identification, we need the fragmentation data. Now they regret. Yeah? How can I get the molecular ion with the soft ionization and also together with the proper ionization technique? One of the students mentioned the some Tuesday, Mateus, and Alves, what DIA cases, they want to deconvolute data. Or technically, or physically, technically, they want to do kind of ion trap mass instead of now ion trap mass. It was quite popular some decades ago, some 20 years ago. Now they want to apply to ion trap to get robust fragmentation ion data, which could help for the structural elucidation. And third one is ion suppression. Ion suppression is an issue. Animal cases, without any chromatography, we can directly inject, directly measure spectrum. But ion suppression mass, usually, if you have a very dirty metabolite, you cannot measure directly mass spectrum. Why? Because ion suppression. Ion suppression, there is a joke. If you have a dirtier compound, you will get a cleaner spectrum. Because one major signal suppresses everything. So, it's a simple word. We want to increase the capacity of mass spectrum. And last one is adapter ion tester. One of the important information in LCMS, like ESI cases, adapter ion, the ratio M plus H, M plus K, M plus Na cases. This is because among all the mass signal, which one is a molecular signal? That's the first question. Of course, mainly is from soft ionization, is a molecular ion, but many cases not. Then which is molecular? Based on those three signals, we found it. But please imagine, deep sea, or some microorganism from volcano, or who knows, as I mentioned, you are going to next year to Mars to collect a new sample. Then adapter ion pattern is completely different, also like a marine organism. Now, how to apply those four? I don't have any answer, but direction is the fourth direction. NMR case is completely different story. NMR case is low sensitivity. Usually mass spectrum, if you have this one, only picomole cases, since sometimes micromole sensitivity NMR. That's the one direction, this one direction. What's the solution now? Cryoprobe, so-called cold probe cases. You keep your all electricity in low temperature, all electron noise is going down, and your signal sensitivity is increased. That's the cold probe. Or micro probe. You have a very narrow down micro tube. Then it's not because of a concentration increase. Also, it's because of a physical effect 4 times increase. But still, even you are using micro probe or cold probe, cryo probe, still is not comparable to the NMR. And second one is complexity. Of course, NMR, HPLC NMR is possible, but to apply to metabolomics, it takes a really long time. And also, as Van der Laan mentioned at the beginning, so ideally you have to use deuterium solvent. Yeah? Not to use deuterium solvent as a mobile phase, you collect in the solid phase category. It takes one sample takes 1 day, but you have 1,000 samples. You have neither. So that's why still to get popular NMR is not possible. Then you have to measure extract directly in NMR. But I told you, one NMR spectra of a pure compound, one pure compound is more than enough to drive you crazy. But now 30,000 chemicals in one spectra is a really bunch of signal. Then we need a deconvolution data. Then also LCMS cases, molecular weight is very, always I made a joke. To measure mass is more difficult than NMR. But to identify, to interpret data, NMR is more difficult. NMR is now you have a bunch of signals, how to identify. We need a database, actually. But LCMS cases, mass databases, ideally you don't need the data. As long as you have accurate data, you can Google. Or 300 1 to which compounds are coming up. But NMR cases is because all the signal is a pattern. But primary metabolites, of course, primary metabolites, we have many databases, but secondary metabolites. 90% I was there from secondary metabolites. Like today, Van der Laan or Fatima mentioned indole alkaloid, benzoquinone alkaloid. Those compounds, where I can find the database? That's the issue. So, one single method is not enough. So that's why our group, we are using multi-platform metabolomics. One is not enough, then we can use several. So first, always we are using NIR spectrum. Although we are targeting the single small molecule, but many cases, I don't care about the size of this one, like a potato cases, or such sweet potato, main metabolites are those one, those starch or inulin, those cases, we cannot detect in mass spectrum in animal case or in animal case. But I want to know, in that case, a simple method is we can do with the NIR spectrum separate the classification. If you found more in NIR, then we have to go to the polymer analysis case. For example, now you have two potato. One of them is infected potato. One of them is healthy potato. Those are no different in metabolites, but only shows difference in NIR. Then we are barking for wrong tree. It's not metabolites, it's the polymer cases. Second one is we are doing usually NMR, but sometimes HMS, overall metabolite. Because it's unknown plant material, I need a pattern, or I need the overall rough group of metabolites. In that case, we are using NMR. After NMR or HMS, the other way around, cases, now I found the flavonoid or particularly NMR, a terpenoid is changed, then LC we focus on these terpenoid and flavonoid cases. And or if you have find a volatile, probably we can go to GC headspace cases. Always first level is NMR overall, second level is mass-based one, LC-mass or GC-mass cases. Now we applied multivariate data analysis or data to identify the compound, but as I told you, 90% of the metabolites we failed. Now it's a bit increased because of molecular networking from GMPs. But still, some people are interested in new compounds. New compounds mean it's not listed in the database. How to find this one? Then you have to isolate the compounds. Of course, now based on this one, you spend several months only for the metabolomics. Now, if you tell your supervisor, I want to start to find a new compound, then your question immediately by supervisor, what did you do so far? Yeah? In that case, now isolation technique should be fast and simple. In that case, we are using TLC cases. As I told you in my course, if you do TLC, now you can see here, those bands is very characteristic. We can scrape out to the interface. We go to the animal. Then immediately we can solve, or animal case, we can solve to identify the compound which are not listed in database cases. That's it. And molecular networking. So now there are two kinds of technique quite common. Molecular networking here, and toxic. Have you heard about the toxic? Statistical toxic. Why? Because now it's a common trend both in animal and LC-mass to do classification. Now when you look at 300, you don't know whether this one is flavonoid, whether this one is terpenoid. NMR, of course, NMR shows some more example. 7 ppm is kind of aromatic compound. But I want to know which class it is. In that case, LC-mass is clustering cases, like a molecular networking in GMPs. Then you cluster the compound. Okay, 300, you detected. This one probably is similar to lipid or flavonoid. NMR cases, all this signal, we can say is all fragmented functional group coming out. Which signal coming from one molecule? Then statistical toxic is not the hardware analysis. It's a statistical toxic. We can classify, okay, 7 ppm and 1 ppm, 2 ppm in the same molecule, then we can go to this one. Actually, completely different approach, two different device, but their goal is the same. Still, we don't know the classification. And another one is in our group. Of course, multi-platform metabolomics, NMR, mass is good, right? We can, if you have money, we can do both. But of course, else NMR mass is possible. But in between, there is a problem. Yeah, we can combine. You just combine two data? No, it's not true because most of them overlap. In that case, TLC, we are using hardware interface. Now I did NMR. This signal is very high. Then here, this signal, I go to the HMS cases. Of course, I don't have to explain detail. TLC itself, fingerprinting is very nice, but identification interface cases, we can apply it. And here, now TLC, also we revisited, as I mentioned this one, TLC cases, same TLC, we did a different reaction. This one is for the is a ginkgo biloba is for the terpenoid. Ginkgo biloba, ginkolide, A, B, C, J, and this one is flavonoid, and this one is ginkgolic acid case. We combined this one, then you got a very nice separation in TLC. In my opinion, when you do fingerprinting itself, TLC is not bad. It's even better than LCNS or GCNS case. Because fingerprinting itself, although the resolution is not as much as LCNS or animal cases, but they showed very different one. We used the color, RGB color. So one then shows different color. So this is quite nice. And another one is we needed rheum species anthraquinone rich plant material, which means it has been used laxative in China, laxative for your constipation, laxative. But interesting one is these rheum species in Chinese pharmacopoeia, they didn't define species. Rheum palmatum or Rheum tanguticum, both can be used for same purpose. But there are factors, genetic. Usually, if you have any factor, genetic factor is the biggest one to change the metabolites. But this one is interesting one is Rheum tanguticum and palmatum, they are growing different altitude. So that's why we tested whether I suppose this tanguticum palmatum genetic variation should be bigger than altitude, but apparently completely different. Tanguticum palmatum, Chinese ancestor, they knew already. It's almost the same. But altitude, depending on the altitude, quality is completely different. You don't have to worry because in Amazon you don't have any mountains. But my case is a very important altitude. So this one is we use. And another one is starch and potato. This one is one of the example NIR cases. Starch and potato. In Netherlands, we have a very many kinds of potato is here. Now, here, when we do NMR, we can see only broad signal. But NIR cases, you can see very nice. It's corn, potato, rice, starch, and wheat. Of course, it's changing, yeah, polymer. But if you do different cultivar of potato, they are quite well changing these cases. So NIR, don't ignore. Sometimes when you have information, like when you are studying, investigating infection, always, oh, I want to do metabolic. But who knows the first syndrome of defense mechanism to strengthen, lignifying, or starch changing cases. In that case, NIR is quite nice cases. Now, problem is, still, when I use multi-platform or mega-platform, whatever, it's a tip of iceberg. 99% is unknown metabolites. This case is, of course, it's a 2006, I published this one. The colored metabolites I detected in my metabology. But most of black metabolites I didn't detect it. So that's why unknown metabolites I feel with this CAP database. But once more, I want to say, our database of biosynthetic based on pure organism. Yeah, pure organism. Now, life is more complex than you expected. Sometimes there is error. Of course, this one is a dissolution, but we have to consider problem of current application of database, the database of biosynthetic pathway to fill your unknown and identified metabolites. And database now. Database, yeah, everyone wants to database, but no one can fulfill because it's too huge. Now it's a GMPS is a quite nice, successful story. They increase all the data. Animal cases, they starting actually since 1917. But can you find any secondary metabolite database? No. Because they believe they are expert. Yeah, they don't need a database. That is the problem. But LCNS database, rather, the which technique is needed more database, as I mentioned, is not mass. As long as you have a very accurate mass, you don't need it. You can do Google 300 1 to what kind of compound, it's no problem. But animal cases, we need it. Then many people think, okay, we need the chemical of database. For example, 2000 chemical. In my group, I have 2,000 animal database. Good news. If you have a 2,000 animal database, because animal is similar compounds shows a similar structure. Net completely different from less. So that's why even you have a 2,000 animal database, it's working like a 20,000 metabolites to gas classification. But the problem is, still is far to go. So that's why now I'm thinking, if you cannot do build up this secondary metabolic database like ACD cases, why not? We can do metabolic barcoding with the animal spectrum. This one is I present in the Salvador, so it's kind of a preview. Yeah, this database now is here. Why this one is I mentioned in the course, this one, but the problem is not just the measurement like MS and upload. No, we need the processing. Not processing is kind of interpretation. Please look at this one. Please look at this one. This one is narranging cases. Now, if you don't know the information, this one is you have in nature two forms, R form, S form. Yeah, R form, S form. Now here, it looks like it should be double-left, but apparently this one is a two mixture of double-left. Then you can say, oh, this one is an impurity. This is the wrong case. So that's why one spectra you upload it, you have to assign. We call it assign all the signal, and you can upload. This case is now is what's this? It's a detection case. Detection, it looks like impurity, right? Everyone, if you don't have any information about the detection, they said it's a impurity, but it's not impurity because of rotating cases. Once you know, it's very nice information. But once you don't know, you ignore this one, you will lose the information cases. Now, question is here. No, we want to use this animal database for the fingerprinting of species. Now I want to build up animal database of medicinal plants in Korea and China. It's several thousand species. If you have this one, now our case is a problem is we can, according to Nagoya Protocol, we cannot use Brazilian plant material. If you found the Brazilian plant material, I compare in my database which plants which I have in Netherlands is very similar, then we can find alternative one based on metabolic pattern. Then for this one, first requirement should meet. What's that? Metabolic pattern is related to activity, but everyone believes. So synergism, everyone, how to do synergism, everyone believes it, no one proves it. This is a problem. Yeah. Why so diverse? That is the first question. So why plant produces so diverse metabolites? That's strange, right? They are not producing for you. They are not producing for our anti-cancer activity, but they are producing a lot. They said, if they produce one glucosinol, they are using 10% of metabolic energy, which is very expensive. But some plant material like pyrrolidine alkaloid or glucosinolated cases, they produce a lot of diverse compounds. Then my question is, my answer is, to draw the conclusion, please have a look at this one. This one is Sardinia in Italy. And I collected the Cistud Montpellier's plant material, very different places. Each place I measured it. It's not, we didn't test it whether it's a genotype or not, at least it's ecotype, same species, by the botanist, same species, I collected the plant material, and I did it metabolic profiling. And then I tested antifungal or antibacterial activity. Then, as you expected, activity is different. Why? Because their environment is different. So, according to environment, they produce very different compound. That is everyone expected. But now, I did metabolomics. Then, which metabolites are related to this one? Strangely, flavonoid. Flavonoid shows related activity and terpenoid. But terpenoid cases is not very the same compound. Skeleton is a diterpenoid, but very different. So I have to prove more, but there are hypotheses coming out. Flavonoid, every plant material, every cistus plant material collected from Sardinia, they have the same flavonoid, which means it's a pyranol. First, pyranol and triterpenoids, diterpenoid is kind of a specific compound. So after curing flavonoid, and then this terpenoid, probably it cures specificity cases. So plants, they have a very complex system. Sometimes it's smarter than you expected, but that's sometimes always. Another interesting experiment. Do you know this one is jasmonic acid? It's a very famous plant hormone, right? It's like a Friday afternoon experiment. Okay, this jasmonic acid, like a plant, they are producing a lot of metabolites. Yeah, it's like a signal molecule. Looks this one. Yeah, this is a simple question. We did it, but here jasmonic acid here. Now those treated one, you see, now is increased. This one is non-treated one. Then we isolate the compound. It's simple. Even here, this structure, anthranilic acid. We were disappointed a lot because we believe after treatment, jasmonic acid, a very complex structure comes out, very simple compound. Even we isolate this compound, we treat it, there is no activity. Then story comes out. New story comes out. Probably plants is really smarter than you think. Now, plant does not have to make that increase, but this plant can see synergism. Now, plants produce some signal molecule to the microorganism together with the existing metabolite. Activity is changing. Then we did it, these compounds together with antibiotic, then activity is increased. That's a synergism cases. And now isoflavonoid. You know, isoflavonoid is one of the famous phytoallergen cases. 1970, this one is a hot issue. As I mentioned, still that time we believe plant does not produce antibiotic. But as you remember in ecology, some people found it antibiotic, this isoflavonoid is completely very increased by the infection of bacteria and fungus. Many scientists were shocked. Okay, this one probably is the first new class of antibiotics from plant material. But situation, life is never been in my way. Yeah? When I isolate this compound, activity, zero. If any, it's very mild. So that's why they give up. Then they use just the compound. But 2017 or 16, we tried different way. We isolated all the isoflavonoid, what we bought is isoflavonoid. We combine activity together with the current antibiotic, like I forgot some band, the tetracycle class of the compounds, those things, activity with this isoflavonoid is getting increased. Now it's sometimes 10 times or 20 times increased, which means plants, as I mentioned, it's like hypothesis. A plant does not have to do direct antibiotic. Instead, they can produce isoflavonoid to help their friends antibiotic agents. One of the examples is a commercialization. It's not our result. In South Korea, the Peporine or flavonoid, together with omeprazole, they are working very well against the Helicobacter cases. So flavonoid, many people, antioxidant, they want to develop a new drug, flavonoid, but most of bioactivity flavonoid is very mild, so they are disappointed. Probably new direction of flavonoid might be in a synergistic to increase the synergistic effect. Another advantage of this one, this isoflavonoid, always lifespan of antibiotic is very short. After some years later, because of multi-drug resistance, we should ban tetracycle compound. But when you combine with this antibiotic, so isoflavonoid, probably we can reuse this tetracycle, so banned antibiotic cases. So interaction cases. Last example, a few minutes. Yeah, everyone knows that our group is I'm famous natural detected solvents. You heard it. Actually, this one is the best outcome from metabolomics. Now, you learned it, phytochemistry. Yeah, phytochemistry. Among the 30,000 chemicals, 99% is not water soluble. If you isolate the texture or alkaloid or flavonoid, are they soluble in water? Never. But the strange is, you know, the organic synthesis. If it's not soluble, we believe only media in nature is water, right? But strange. 99% is not soluble in water, but they are synthesized in plants or in microorganisms. That doesn't make any sense. So we started this one. Probably then plants might be another media. But unfortunately, ethanol is only from cachaça, not from plant material. This is a problem. So that's another one is resurrection plant. Yeah, Selagnia plant, resurrection plant. When they are grow in the dry season without water, they still keep survive. But DNA is okay. RNA cases without water, they are very unstable. Yeah. How do they resurrect? How do they keep their lives? In the desert, or even if it's minus 80 some plant material, they can produce, they can survive. There must be unknown media, right? You don't have to agree, it's already published. Yeah? I don't need your agreement. Yeah? And here, another one is this one. So I collected from, it's a carnivore plant. Carnivore plant is not only insects, sometimes they catch the birds. But please think about this one. This one looks like water, right? If it's a water, it's big problem because under the sunshine, so particularly in Brazil, everything is within seconds, it's dry. They cannot catch. There must be another liquid present in the plant material. Unfortunately, I was working at that time in TU Delft, technical department, which is nothing with a natural product. They were working synthetic ionic liquid or dipeptic solvent, which means if they mix up two solid, they become liquid. They were using a lot. Well, when I look at what they are doing, probably I always worship my nature. Nature should use this one. So that's why at that time, I had all the animal spectra of several thousands of species. I checked it, metabolite. Interestingly, this kind of a compound, of course, this one is primary metabolite, always is higher. This kind of also Friday afternoon experiment, I mixed up together. And then they become liquid. If you mix up malic acid, which has a melting point around 120, and together with the sucrose, which is, I think melting point is around 160. If you mix up, you dissolve in water. Of course, it's not mixed up. Dissolve in water, you dry the water, they still keep liquid. Strange. You don't know? Then, one of the hypotheses, proven hypothesis from textbook, do you know vacuole? It's a vacuole. Vacuole is kind of a trash bin. So all the toxic chemicals in vacuole. But vacuole's problem is water content in vacuole in rainy season 90%. But in dry season, only 10%. But can you imagine toxic chemicals in 90% is well soluble, but 10% in dry season, it crystallizes, plants will die. But I never heard a plant is dying because of actual crystal. But when I checked the textbook biochemistry without any clear explanation, sucrose, malic acid, the second environment metabolite to water without any clear reason. But when you look at this one here, sucrose, malic acid, when you mix up, then they become liquid gases. So then here, my story is in the dry season, water is here, dissolved. And then they are getting water, rainy season, and getting reduced. Then sucrose, malic acid, getting concentrate, they are doing as an alternative media. That's a story. Always I better to be a story writer instead of scientist. And another one is routine cases. Do you know routine? It's one of the famous routine. Routine is sometimes so far as a plant material, dry weight is 60%, 40 to 60%. Can you imagine non-water soluble compounds in the flower part of Sophoracea is 40%? How is it possible, you ask chemists? Not soluble water. And also some metabolites synthesized in root. And then 10-meter higher square plant material, how do they transport if they are soluble, if they are not soluble in water? In that case, we tried several nades, then its activity, solubility is much higher than water. Actually, water solubility is 0. And another one is nice, is an example. I told you, plants without water, they keep their life. How is it possible? So that's why I put one of the simple enzyme lactase, and nades is here. I put it. And then I dropped one drop by drop, water. It's like mimicking raining. Then now 50% and 25% and 50%, then enzyme is activated, which means in the dry season without water, they still keep their life. It's not denaturated. Now, actually, I started this NADES project as this kind of very pure scientist academic cases. But now, and you check it, the Google NADES, every day hundreds or thousands of paper comes out, comes out, and then many people are interested in the application, like green media for drug formulation. And I have a patent for cosmetics, which was bought by Dr. Fosse, French one. They are paying royalty to this one. I was lucky. No? But this one cases, those one is very nice cases. So now nature, once more, is the source of bioactive compounds, but also the treasury for valuable system. Learning from nature, applying to human. Everyone knows it. We call it blue technology. This one could be done by metabolomic cases. So then what else? We are different. Maybe because this one is many students comes here. Now is I formulated some example which you might be interested in. You know endophytes, endophyte and gut microflora. Gut microflora. It's completely different, but when you look at the mechanism, they are very similar. Plants, gut microbes probably is endophyte. Endophytes probably is gut microflora in plants. Second one is latex. Latex chemical composition is very similar to your snake venom. So if you want to study, maybe there is a correlation. And second one is plant peptide roles. Today, Van der Laan already mentioned peptide. Plant peptide cases is a it's not very well studied one. Plant peptides is rather a mammalian cases, but still, these peptides are not protein cases. We have to study, and the Laan shows the example as of a systemic case. Not only active compound, what I mean is in the system. And volatile. Volatile, as I told you, is not only for your perfume. Volatile is the SMS, text message, or email between plant material. Probably, then, many people think, okay, one volatile can deliver one message. No, it's not true because we have a very limited metabolites, but I have to send all the messages, love letter or warning letter or angry letter, combination cases. This combination is kind of what I believe is kind of decoding someone. This one is a scientific fiction. I want to give some example. And last one is now reverse. Pharmacokinetics in Mars or Moon. We have to, one day, who knows, Tesla guy, he is sending a spaceship. We have to prepare for the plant's material in Mars. No, organisms in Mars or Moon basis. So now 19th century, 1900 is chromatography, antibiotic, 1930, 50, gene, what's the name, Crick? And spectroscopy by Ernst, and 2000, everyone knows it, omics. Not metabolomics anymore. Now it's big data, AI is how to apply. Everyone knows it, but AI is not automatic robot. You have to find where to apply these cases. And this one is, of course, it's not only my work, all my colleagues on funding source. Thank you very much.
For question because we don't have the next lecture. They have time for question.
Actually, best lecture should keep the time. I always keep the mode, but today I heard someone cancel, so I used thin mode kind. I'm sorry. Yeah? As you know, in the lecture time, I always keep very nice, 12 o'clock exactly.
I love one of your last messages about the necessity to understand the nature to apply for humans. It's exactly what I try to do. And in your case, because I was the initial story, because I was traveling, and you're looking only to understand the physiological function of these small molecules in plants Or you have also research between plants and animals and things like that?
So actually, so first, of course, plant is, but as you see, I always want to use plant microbiome interaction. And also plant, as I mentioned, so in the course I mentioned to the student, flavonoids, we believe there are many roles for pollinator or UV protector, whatever. to me, it's like a flavonoid, the plant microbiome interaction, whatever, is kind of a sunglass of plants. Because a lot of light condition is here based on combination of these flavonoids, they can observe what they want wavelength is. With my colleagues, actually my neighbor is, but the snake cases, I'm collaborating with the snake guy, the upstairs. And I'm plant chemist, but next door I'm working with a soil guy of business. But as you mentioned, plant is not only plant visual interaction now is more. Yeah.
My question is, maybe you.
But don't forget, I'm sick today. Yes? Don't, please treat me very softly.
My question, I think, is just you think about. Uh, you talk about marine organs.
Yeah.
And marine organs, you can find the sesterpine.
Sesterpine, Sester 25, C25. Yeah.
Yes. When we started with Gottlieb, working with biosynthesis and learn about biosynthesis, we didn't have sesterpine. If you look for very old books, there isn't sesterpine. Then, if you believe in how the evolution of the life, and if you believe that the life started in the water, then some organism came out, then you can start some forests, some plants. And frequently I think about probably some genes for the pathway of sesterpine was silenced. But I don't know. Every congress I go and scientists talk about marine organisms, I do the same question. Out of the Brazil, for a very famous scientist, anyone discuss with me with evaluation. Do I have some idea? Have you read some papers about it?
Yeah.
Why do you think about this?
Basically, I don't know. That's the that's it first. Let's study this. But there is a student who follows the mind course, but I want to give a very related answer. Is that okay? Some people say when I'm always fighting with the microbiologists. Microbiology, they said it's like nowadays general natural product. Microbiology, when they isolate the compound beautifully, they always start from gene cluster, gene and metabolite case. But when you are doing plants, gene cluster is impossible, right? Gene is everywhere, so it's not clustering, it's cases. So there's a with the connect gene, it's very different. But her question is C25, which is in terrestrial plant material, very rare plant material. Question is, in marine organisms, do they have a new enzyme or gene or not? Or my case is, instead of a new enzyme, as I mentioned, marine organism is always is clustered between two organisms, three organisms. It's not like plant material or microorganism. Probably two possibilities. They are helping each other, like endophyte plant cases to help to synthesize one compound. Second case is maybe like a triterpenoid today, Van der Laan mentioned indole alkaloid, two pathways, tryptophan and secologan cases. When you do plant tissue culture always we fail. Why? Because localization, they are located in different places. But in marine organisms, who knows CF and G5 in the same places they produce. So, to know synthesis by new genes, fine, can be possibility. But if you want to study, there are always possibility is localization. Second one. Third one is synergy between two organs or multiple organs. There are three possibilities. Once more, I don't know.
But anyway, I think as is, I have more questions. so that we can discuss. But I would like to do this question for most. No, you know about the question. You say that why so diverse metabolites we can find in plants. I disagree because we have some family in the you don't find anything. The family is very poor. And it's common, the literature say, it's because the plant has condensed tannin. And they don't give any other defense. I don't in my opinion, I think you didn't have a condition, good condition to work with these plants. And in my opinion, we need to work again. We're starting to work again here with a group.
Even same plant material, depending on the condition, they differ. But my basic idea, plant why produce very diverse compound. One big, the level is, they want communicate positively, negatively with the unlimited factor with the limited number of compounds. In that case, only synergy or combination is possible. That was my point. I still agree with you.
I have to say with you.
Yeah.
I don't believe that.
Yeah.
Don't have compounds like that. Just the way that you
Like as I mentioned, so some people mention like God microflora, let's say volatile cases. Volatile, when you look at many volatile like a pine tree or lemon, whatever, actually there are, if it's a compound from gene, there are two possibilities. Or it's because of environmental cases. But many cases, it's not only from the volatile cases. I think its case is environment is more influenced of this one because they develop this to communicate with each other with a limited number of compounds. Probably they want code like peanut, whatever. This one is this message, this kind of like our nucleic acid coding cases. I believe this one is scientific fiction. Maybe depending on the one of the paper is Alvaro Fleur in South Africa. He mentioned these cases like this volatile. Instead of individual profiling, pattern is more important related. He mentioned activity, but I mentioned for the physiological role. So today, not 100% my description or explanation is correct, but only to open your eyesight. This is my.
Okay, let's sing again, Professor Choi. Você não atende o Norberto aqui para ver onde ele vai pôr.
Você tem pegado, professor? Deixa eu quebrar. Posso usar o mouse?
we have a lecture from Professor Norberto Peporine Lopes. He's going to talk about metabolomics in chemical ecology. Professor Norberto Peporine Lopes is a pharmacist, master in pharmaceutical science, PhD in chemistry from USP University in São Paulo. He's currently a full professor of Faculty of Pharmacy Science of Ribeirão Preto, University of São Paulo in Ribeirão Preto. He was president of the Brazilian Chemical Society. He developed his postdoctorate in mass spectrometry of natural products at the University of Cambridge in UK. He has received several awards with emphasis on the PR Mass medal. It's a Brazilian Mass medal. He works in the areas of natural products chemistry and mass spectrometry with emphasis on chemical ecology. Exactly about the title of his talk today. Please, Roberto.
Thanks, Fátima. Eu não preciso de microfone, não. Sai a voz aqui. Sorry that I saved the talk here to transfer. I lost the title that I was typing. It's a bit different, but it's the same idea. First, I want to excuse that I was not here this morning, today, in the beginning of the conference of Professor Choi, because we have the meeting of the Academy of Science of São Paulo State, and I'm organizing the meeting for this afternoon. I need to give the talk and escape. Sorry about that. I move in this one. Yes. Just in the beginning, I want to show you a short history where I am and why we decided to talk and work with ASCAP. I did my undergrad in pharmaceutical science, as I told you. And when I returned to the same school, to the same department, to work as an assistant professor, a long time ago, I was included in a group that we call NPPNS. This group here worked as a nucleus for a long time. The group started in 1966. We still work in a way that everybody has the money together. We share everything, like you have it here. And in this group, when I arrived there, the main idea was to work with Asteraceae plants. And I was the first one who rebelled a little bit this idea, because I was one of the first in the beginning who asked to work much more about the relations in the nature instead of to do chemistry to identify new sensitive lactose. That was the main subject. Today, the group, the nucleus, has people working synthesis, marine organisms, cave microorganisms, different kind of subjects. And that's one point that made me thinking about science for all my career. I draw this picture in 15 years ago, something like that. But this original draw, I did it during my PhD after some class of Professor Otto. And as Professor Choi just told us, it's, in my point of view, it's impossible to think in a plant without thinking in all environmental, because I will try to show you that different conditions, different interactions can induce different answers. And the plants, at that time, for me, was the only way to interact was based on chemistry. And that's the reason we start to work in different kinds of chemical ecological interactions. Today we work with frogs, we work with bees and other insects. We apply some of the concepts that we developed through the gas phase in humans, and more recently with monkeys. And what we try to do, we still work with plants, but the idea was to look for different interactions. How these monkeys eat some leaves, when, if it's for protection, it's for healthy, or for, doesn't matter which the intention.
why some bees go to some plants, collect different parts of the plants, why they go to that one and don't go to the other one, and try to understand, or in the case of frogs, how they interact with between the frogs and the microorganisms through the chemistry. And the idea is, if I start to work, in my case, with more the biologist guys, try to looking for the nature, probably I can find something that can be useful for humans. But in this start point, we decide, after my postdoc, to apply concepts of mass spec in these interactions, first with plants. Okay, in this proposal, I started applying the knowledge that I get during my postdoc in Cambridge. Professor Stoughton, he's retired now. He had a company at that time that called Bioactive. Today, one big pharma bought this company, and Stoughton is enjoying the life half part of the time in UK and part time in Chile, in the mountains. Good guy. And the proposal was to modify microorganisms to produce tons of mutants and use mass spec to see if this mutation can afford new molecules or new derivatives for some commercial molecules. And to do this in a very fast scale, they applied at that time mass spec. And I start my background in this group in chemistry, but as Professor Choi told us, you can use, I think the battery is gone, you know. You can use mass spec to see the molecular signals. For example, protonate molecule, deprotonate, sodiate molecule, so it doesn't matter. You can look for the fragments. And in our case, what we love to work is with the gas phase chemistry involved in this process. And then I was learning how to work with computational chemistry to define the transition states and all these steps in each single reaction. And then we start to building at that time some maps for his group to monitor at least ionophores. And the idea was, okay, let's back to Brazil and use this to understand initially plants for laceration. But just when I arrived, we had a very dramatic problem in the hospital in the University of São Paulo. Probably you don't know, but several people who had kidney disease in one day was in contact with star fruit. And star fruits, for people who has kidney problems, can induce several answers that in the end the patient can die. And this is a long history. I don't want to go in details because I have 30 minutes. But if you see in the animals, that was the idea at that time. To do bioguidance. Just to give you an information, the end of the history. To isolate the toxin, it was necessary at that time 20 kilos of lyophilizate star fruit to get 2 milligrams, which means you need a small truck of fruits to have 2 milligrams of the compound. And these guys were doing in US and China, that was the two big groups that were working in this subject at that time, and doing animal assay. I don't want to show the movie, I prefer to show the picture, but with the picture you can understand how terrible it is. They introduce directly the toxin in the brain, and we get some information about these groups, about possible mass outbreaks. Then we did it the opposite way. We start to build small platforms to check for mass spec. When you have some reports about any of these accidents, we start to do a mass, and we propose at that time, 2013, to use mass spec as a guide to do isolate. You get success. That was the toxin that we isolate for the first time in our group. The people from group decide to call as caramboxin, the toxin for starfruit. The starfruit in Portuguese is carambola. I don't like to word that in the internet. They call this as Brazilian toxin. Brazil is not toxic, come on. Could be toxic with these days of politics, but it's gone, I hope. And it's interesting because this molecule is not very stable. And if you have the civilization here, you lost completely the actives. What was good for us to understand, first, why the juice is not active, because in the juice you don't found the toxin, but you found that one. Water and vegetation. And because this distance, this molecule is exactly like the botanic acid, another toxin who blocks gamma receptor. We published this data, but then we were continuing this research. This is an unpublished data. My idea was in the past to say, come on, why some starfruits have, why some fruits don't have? And normally microorganisms modify amino acids, because if you look in the structure, you can call this phenylalanine or tyrosine derivative, depend of your eyes. Some guys say, no, you cannot say tyrosine, or you cannot say it, but it doesn't matter. What happened here is you have a 6-member ring here and an amino acid. And then the other modifications is done by the fungi. And then the proposal was that these compounds could be produced by endophytic microorganisms, not from the plants. And that's the first image that we get from the microorganism producing toxin. And as expected, the microorganisms were in the seeds. Usually they prefer endophytics from fruits, because there's a lot of water and sugar, and they are more comfortable with more fat and things like that in the seed. Not seed. And there are some things that I really like. This is the letter of one of the patients, that a lady who don't lose her money just because they found the paper. Because as common in Brazil, when the Brazilian scientists publish something that the people don't trust, they prefer to looking for outside data. And at that time, the medicals in Brazil, they call Professor Miguel, the head of the nephro in the hospital at the university, as Dr. Carambola, because they said, no, this is nothing. It was necessary that the news appeared in Science in Germany, the TV in Germany, in the US started to do programs about this, so the people start to believe. And what happens is usually the people, the patients or the relatives of the patients found this in the internet and go to the doctor and say, please treat my mom in this way. But the good point is, when you understood all this, first you can help people. For me, this is, in my point of view, the most important paper in my career, because I can help people with this information. But then we draw the synthesis and the complete process to produce a new molecule to induce epilepsy in animals. For what? To kill animals? No. The people who work in this area for epilepsy, they need compounds to induce epilepsy in animals to try to treat the animals. Then the patent of this new process is now transferred. It's now in the process of to transfer to a company to start. That's just the beginning of the history. I want to call one point. We talk a lot in Brazil to find biological compounds, and we must do this. I don't have any doubts that we need to do. I do this looking for the ecology. But one point that we never think about is, when you go to the politics, to say, try to convince that we need to preserve our biodiversity. What they understand, value. And probably you don't know, but to do value calculation about forest of biodiversity, there are some equations. In many parts of the world, we can do this very well. But in Brazil, we have a problem, because our biodiversity is so high that the equation will define this process that's Monty, not here, Montgomery, they using this equation to divide the number of species by the research. If you take the number of species that you have in Brazil by the number of papers, research, it's almost nothing. And then we are not allowed to calculate the biodiversity value in the equations that the people use in the world. Then, much more important in Brazil now than find a new biological typology is to publish data about our biodiversity. This is really, really important. Then if you have something that you don't believe that's a very nice paper, but it's okay, have an answer. Sometimes a bad answer, it's an answer that can help someone. Please, publish. Our forest will be very thankful for you. Otherwise, I just arrived from Amazon. Manaus was smoke everywhere. We couldn't see the rivers, the Solimões and Negro River mixture, because it was completely smoke. Then I need to improve. Okay, this is another point of view where I want to show you how mass spec or metabolomics tools can help us. Because in Brazil, to keep the biodiversity where they must be, it's important to show some values, something for the politics. And that in U+20, with Professor Jolie and Professor Van der Laan, we propose one strategy to don't look for a single species, but to try to look for an environmental complete one. Why are we doing these things? Why I propose this? Because if you want to keep this, if you put fence here and close everything or build walls, you cannot protect, because during the night, the people who live in this area will jump and cut palm trees and kill animals because they need to survive. They have sons, they need to eat. Then we need to, if you want a conservation area, you need to think how to conserve the people who live there. And the way is to what? It's not here. But if you go to the United States, for the ONU strategies, one is to improve or to reduce the poverty, to give money to these locals. Then the idea was, okay, let's think about plants. What I call poor plants is plants who doesn't have many different compounds in extract. Why? Because if I have a big mixture, I cannot make a small factory for these locals to produce something. That's the idea. And we can use, for example, mulch, because it's very fast. You can do like 400 samples in 6 hours. What's the idea? You have this in the aquarium, collections a lot. You can prepare samples for an area like that, where the people from the botanic from New Zealand has already classified all the plants and shot everything. Just shot. What's the idea? Let's shot leaves, because I don't want wood or roots. Shot leaves. And see if you have some extract that it's enriched for something. Doesn't matter. Doesn't matter. And if I have things like that, I can look for them. Any bank, go to molecular formula, and then if could be something interesting, you go for structural elucidation. And if after this step you have an important compound, a key compound, you can go to the government and propose this protocol. I remember when I showed this idea in the conference from American Chemical Society of Pharmacology, and during my poster, some guys coming to me, start to laugh, and then call me as a jungle boy. I said, okay, it doesn't matter. But then we decided to try, because at that time it was just how to get the data. And in the first, this is the crude extract of one leaf of this synthetic. See? This is the extract, just one compound. Do you know what this compound here? It's a precursor for the alkaloid. That's the value in the market. Then we propose a factory for the government in São Paulo. That's very easy. It's just water crystallized directly. And this was elected one of the examples for this conference here. And not in Brazil is working, but in Panama now they have it, one of these forest programs, one of these actions. I'm going fast, but this is what happens when you think in this way. Another way to use metabolomics to find something and talk about it, metabolomics to looking for this support for people in Brazil and also understand the ecology in both cases. Everybody knows I really agree with you. The only difference is I call this terpenoids as notes in music. Well, if you only notes, you don't have music. And every time when you play in a different way, the notes, you have different kind of music. Your example later, we are thinking the same way. We do a lot of this in chemical ecology in our lab. And Professor Lina, she was involved in a program to kill Aedes aegypti. And she invited us to work with her to develop something to use in Brazil to treat water, to kill these insects. The compound that usually are applied in Brazil, stebofos, is very toxic. It's horrible for the ecology, for the environmental. And then what I told her, okay, I want to work with very simple compounds, and if you need a product to show to the Minister of Science, you need something that can scale up. And it was my decision at that time. Then what we did, we're looking for tons of oils that we have in Brazil. This is the GNPS for DCMS. I don't know if you are using it. It's amazing. It's working now very well. And the idea was, it's easy to do the assay, the biological assay. Let's send 300 samples, 300 different kinds of oils and things like that to the assay, and then put everything together. And see the GNPS if you have some molecules that appear and do not appear in active and non-active extracts. And then, in the end, some compounds start to be bigger in the nodes and branch, which means that every time when I have one extract that's active, I have these compounds inside. Then, okay, let's go to the compound now, because this is a very fast way to find something. We bought, because it's commercially available, the molecules, the biological activity was amazing, and this is the products that we offer to the Minister of Science. Now, in Brasilia, they have around 3,000 of pills spread in the city, doing the first big experimental process in a city in Brazil. I think this will be spread to the other cities. And we did everything in Brazil very fast. We start from the TRL1 to 9 in 2 years. Another way to use mass spec to find something active. But looking a bit more what we can do with mass spec in this way. I was working in my PhD with Professor Masayoshi, my co-supervisor was Masukato, and at that time we published the information of this pigment here as working against Chagas. Okay, it was, we tried to do a patent, things like that. When I was young, I didn't know the difference between innovation and discovery. This was a discovery, not an innovation, but I learned now. And okay, we continue this. We work with a company in a FinHab process to understand why in vivo, sometimes this molecule was really nice for Chagas disease, but sometimes it's not. And trying to discover other possibilities. What we did, first one, this coming from a leaf in a plant from Amazon. You can obtain, you can do by synthesis. But then with Masu, we did what? Plants from Piperation sometimes store these molecules. We started looking for plants who can have granzine or veragencine, doesn't matter if no metox here, both are biological activity. And we found one extract that you can directly crystallize. Now I can do to clinical trial. Because now I have a source in big amounts. After this step here, we start to be close to innovation. Before it was just discovery. And then I was with this question in my brain. The project with Licknoff was to move, to forget for Chagas and move to Leishmania. And then what we did, we used computational tools to draw images of the molecule in the animals. Why we do this? We vectorize all the animals, the rats, and now I know why it works. This is the first image for a natural product, the pharmacokinetic, the distribution in the complete tissue. See, if I have the Chagas disease here, in this part of the muscle, I can kill the parasite. But if I have in that region here, I don't have distribution. The molecule don't spread with the pharmaceutical formulation that we had until now in the heart. On the other hand, we keep in other tissues in high amounts. That's more than necessary. Then that was the image. What it's necessary if you want to think about this. And I forgot to say, I don't have here, but with the leishmaniose, you have the product. We did all clinical trials. We are waiting for the human authorization, and then in animals, we have the image that the formulation with this molecule for leishmania, it's really beautiful, but it's in the patent document that we can have success. In the same way as you told us, Choi, the first data that we have with this plants distribution was with Licknoff. We collected Licknoff for different areas, but from the same environment. Then I have a little modifications. But Professor Otto teaching during his class a long time ago that when you have a plant for one biome that is growing in a border between two biomes, these species will have interactions between both biomes, sets for both biomes, and they must amplify the chemistry, the fence. What we saw, this is the typical Campus rupestres. All the Brazilians arnica grow in this area and have this distribution. And here is the CS13 lactose. But when you go to the border between a Campus rupestre and one forest, like the Atlantic forest, you see a completely change. We amplify the toxic compounds. You keep the other ones, the flavonoids and the chondrogenic acid derivatives, but you amplify. And the funny thing is, if you take the seedlings from here and put here, and the seedlings from here and put here, you will see the change of the chemistry. Which means, who defined this chemistry? The environmental. Then if I go to look for anti-cancer compound here, nothing happens. If I go to antioxidant, for example, that I expect to have, both will have. But if I go here, and one of the minor compounds of this area was amazing active. This is the animal with a kind of cancer. This is not published data. And after the treatment with the molecule. The funny thing is, we can take the mass spec and try to find here as a minor compound. You don't see any biosynthesis in the other point. But I was interested also to understand why one specific C flavonoid occurred exactly in all the plants from this genus. And also, for the other genus from Asteraceae in the same environmental, we found the same molecule, not the class, the molecule. For some biologists, when you have this, it's a kind of conservative metabolite. As I told you, I work with the GNPS from the beginning, and we apply the GNPS to understand what happens with this flavonoid chemistry. We do another strategies, but what I like is the gas phase chemistry. Then we did a lot of research to understand possible difference between the isomers of the flavonoids from Echinophylla, because we have one that happened in all the plants, but in the same family, we have other 3 molecules with the same molecular formula, which means we can do a lot of confusion. Then don't go to the details here, but we define what we want to have, and that's the molecule, that's the major fragments in MALDI, because as expected, in the MALDI, the fragmentation is not exactly the same as the electron spray, because you absorb the energy from the aromatics. The internal energy of the ion is not the same. And that's Professor Semi, who passed away 3 years ago. He was the most specialized botanical guy in this family, the Asteraceae. And during all field works, see just that molecule work in all plants that we analyze. The other flavonoids sometimes appear, sometimes not. And he was all the time saying, in this environmental, specific one, with high amounts of aluminum in the soil, with the light intensity that we have, the only way some plants, because just few plants go more than 1 meter in this environment, have this condition, it is necessary to have a chemical barrier. Then we decided to try to understand a little bit. We did the GNPS for different families, blah, doesn't matter here. That's the point. When we found and classified all these 50 or 60 different flavonoids, just listening to the one that happens in all plants has a specific transportation to the top of the leaf, which means that molecules on the other flavonoids are random distribution. That molecules here works as a chemical barrier. And that was the first paper who showed this. It's good, but how can I apply this knowledge for make business, think like that? The first point was an observation of one of my ex-students that she realized that some bees go here to collect this area of they go to the flowers, but also collect parts of the leaves where we have these compounds. And we start to understand more about this. And this is the other bee that visits different Asteraceae and collect the flavonoids. And we apply all these gas phase chemistry to understand if they keep intact the flavonoids or if they modify. And we learned that some molecules they put in parts of the nest, exactly where in the way that they collect. And in other parts of the nest, they metabolize this and store it in another way. And then we define all this chemistry, and the lady was very happy with the answers. And now she has a cosmetic company who works specifically with this kind of things. And she's learning that it's important to know which kind of plant the bees visit to define the high quality of the products. And also it's very nice to teach kids about science. That's the lady, Juliana Ferris. And going to the end, sorry, I was trying to keep here the idea, but it's not, as you told me, don't work all these slides. But what we do a lot of now, it's combine electroionization, electron spray and MALDI, and work with metadata to combine all these informations together. One example was the application in marine organisms with Leticia. We took two different animals from different areas in Brazil, and like we saw in the plant, both species here, both species here, both species here. If you combine, build these files in gold bars or not, if you use the GNPS or not, doesn't matter. In the end, when you combine all the data, you see that two different species has the chemistry more close than the same species in different ways, which means, again, the environmental is driving the chemistry. Then we did a lot of analysis to see if it was microorganisms. We did transcriptomes, things like that. Nothing happens. And you are learning during that time that some animals that live here by the coral barriers, like that ones, can have amazing plasticity we decided to compare and to build metadata, how we can mixture the chemistry with the marine conditions. And what we learned in the end is the animals don't have different skeletons. What they do is they can, in different environmentals, different temperatures, different source of oxygen, they increase in the same skeleton or in the same molecule the number of the oxygen atoms in the core. If you go to more warm and rich oxygen waters, the same skeleton can go to 8 oxygen atoms. And this is new. No, Professor Roth proposed something like this for evolution of trees in the 70s. But it was the first chemical demonstration. And the final example, it's about the frogs that we try to understand in the nature, how they interact, and if for the molecular core. That was, this story is too big, I don't want to go in all detail, but this is a frog, a likely frog. During the night, you can excite if the moonlight return, you can see the animal, it's amazing. And we realized that these animals, they keep the glands with a lot of bacteria. And it was funny when we started to check, because I was not expecting bacteria, because frogs produce peptides who kill bacteria. But in the glands, we found the peptides, and you find a new class of aromatics, a new class of natural products from nitrogenate compound. Then we started to understand all the proteins, things like that. We published a lot of papers, different journals. And in addition, combine everything to understand, because if they can lie to you, they have one way more to increase the reproduction, to be more efficient. Probably don't know, but in frogs, you have the male, the strong one, simply very strong. But you have some ones that you call satellite males that lives nearby, and they do fake sinking. Try to convince the female that he was sinking or it was sinking, and to have success to couple. But when a female can see the animal, it's another one, because you can see far away by the light. But then we took all the family and started to understand what happened with the pheromones also, to understand the complete process. And the funny thing is, the pheromones are a class of molecules that usually microorganisms produce. Try to understand, that's the gland, a lot of microorganisms. And then we did imaging, and we saw that where I can find the pheromone, in the same place I have the toxic peptides, and I have the new class of nitrogenate molecule in the same place, which means there is something going in the soup. And then we did all the transcriptome to define. We found 2 new species of molecules that has resistance with the antibiotics, and if the movie goes. When you isolate the bacteria and put the antibiotics, the new class of nitrogenate molecules, they grow. They use the secondary metabolites as a source of nitrogen. The peptides, they don't care. And what they release? The pheromones. Which means that was the first completely symbiotic process that we could characterize in the museum. And in the end of the history, we learned with the peptides, we asked for some colleagues, one is here, to synthesize for us derivatives. And now this, I lost everything, sorry, from the computers. The idea was to show you that these new class of antibiotics are very strong in clinical trials from the resistance bacteria that we're isolating in São Paulo. And the frogs also are amazing models that we use in our programs to teach science for children. Keep this usually at least once a year in our lab, because I really believe that we need to change the world through the kids. And I want to thank all the collaborators who help these data, I hope I can convince you that it's important to use mass spec as a tool, and mass spec is just a tool. You need to know this. Don't choose in the first annotation because this is not real. And also thank my family to give me all support. And thank you for your attention.
Have time for a question.
First, thank you very much for your very nice and very clear talk. My question is to know about your opinions when you take the data, not only the mass spectra data, but when you take the data, doesn't matter the technique, you take the data, you put all these data in a data bank, in a big data, but different research, different technique, and different things, take the data one different way. How do you trust that? Because most of the people say, oh, this comes from AA.
I don't trust.
Okay.
What I try to teach my students is don't trust it the first time. Because they just offer for you an opinion. That's the reason we call annotation. The same thing for the end. Doesn't matter the technique. Then the second step is work with your data. For example, in our case, we use the GMPS. The GMPS is amazing to organize your data. Then it's just yours, because they are organizing by the neutralization depth. They recognize some files, they calculate the quotient, things like that, to try to propose you something. But what they do is the network, what's amazing. Remember, if you have a flavonoid, for example, if you have an acetyl flavonoid in a GMPS, they will be there, and the other flavonoids will be there. They cannot be also in the same part of the organization. And what I do in the lab is first, in the case of mass, first check the manual annotations. Second, see if
Change form. I have a question.
then you need to back to see if it's a very common, sometimes like routine. Routine doesn't matter. But if you go to the GMPS platform, you see that the same routine for different parts depend on the energy, the analyzer, the way that the spectrum is not the same. But then you have some strategies like the energy, the collision energy, things like that. Then you do the analysis, and if you have one seed, you can spread to all the group.
Okay.
And what I do is, as soon as I have one small doubt, I do the isolation. And the second thing, I work all the time with the UV data. I don't use only MS. I like to use MS with UV. It's a second point. And when it's possible, I do GCMS, LCMS, and UV. And when I'm not confident, I do the isolation. At least of the major one of this cluster. And then, if I have one, I work in gas phase capture. Just because I know how to work with gas phase capture. Just because I like it. But it's not. It's just one way. But I never trust directly. It's difficult to convince my students sometimes. But it's.
Yes, this is the point, because normally, everybody thought, okay, the software showed me this one, but what means the software? The software was, most of the people say, oh, this is a computer. Yeah, of course, a computer program, but the algorithm is very important, because in the algorithm I have a lot of mathematics. And who knows the mathematics behind it? Because if you quest like a simple PCA, there one person in the world that don't know about PCA, it's me. But most of my students can run a PCA. Can press the button. Okay, they can press the button, but is behind, because if you don't use the correct algorithm or use a different algorithm, you have different results. And all these data go to the bigger data, and you have the AI that is saying, "Wow, this is the correct way to think." Don't say that you don't need to use AI, but they care a lot.
In everything. So he told me just before, you have the data. The good point is with the metadata, you can save a lot of time. Save time. It's not the final word. But in our lab, for example, what we do also is building new tools to do NS. Like the last one was the King Walker that we just published. Then we take the GNPS and then take the same data, go to another computational process. We use sometimes 3 or 4. And then you can compare, because now you have 2 or 3 different algorithms. It's a different way to see. One is based on the endoform, the other one is the shape of the chromatogram. Then you can be a little bit more confident, because you apply different mathematical process algorithms to have it. And it's funny, because sometimes the results are opposite. Happens. Because it's like, it's process. But remember, big data will have no more than 5 years, 6 years in it was really growing. You have some things that we did in the past, but not in the way that is. It's the beginning. If you go to the old book for McLaughlin, you see that in the 40s, the GCMS, we had a lot of mistakes in teacher to the fermentation process. And after the 40s, data, 45, 50, that century, they improved a lot. It's the same thing with us. We have a lot of mistakes, back a little, reanalysis, organize, and go one step more. But it's still necessary to check. I don't trust directly.
More question? I have one. That compound, you said that maybe it's phenylalanine, but do you know the biosynthesis? Did you check the
No, I
biosynthesis?
didn't check the biosynthesis. No, they call it this because they classify the toxins from microorganisms the medical way by the amino acid modifications, not from the biosynthesis. Because because the transport for the brain, this molecule use the phenylalanine tyrosine transport.
But it's very interesting because look, it's acetate pathway.
Yeah.
It starts in the carboxyl. You just change the nitrogen, the hydroxyl there, it starts the lithium.
Yeah, that could be direct.
Yes,
Yeah,
yes.
but for to classify the transport,
Okay.
they use this.
Yeah.
But
Because-
those two are possible, actually.
Yeah.
Phenylalanine, if it's from plant, 100% it's a phenylalanine. If it's microorganism,
Yeah,
it's
yeah.
acetate
Acetate pathway.
Yeah.
So it's a plant,
Yeah.
also similar
But
compounds.
looks like acetate. I think it's good to
Yeah.
do the biosynthesis.
Although,
No, we are starting one of
yeah.
the project with the monkey.
Ah,
The
okay.
idea was to do the biosynthesis, but the lady get a position in the Polícia Federal and get a job. Then I need to wait for another one who want to take this job.
but I have a very simple question. You showed the one steroid. It's like active steroid.
The extra steroid, os marinhos lá, fazendo favor, quem que troca aqui? Aonde que tá? Tá aqui.
It's actually I missed your comment about the active steroid.
Probably I didn't.
Yeah, this one.
That's it.
This one is from which organism it is?
In both species you have.
Which one?
That one's.
It's a
Oh,
it's a sponge.
no, it's not.
It's
It's an animal.
animal.
Yeah, it's an animal. It's I cannot say soft chloro because I did it once, and the guys almost killed me. Because growing the same way, I said, this is a soft chloro. No, it's not so. Softer chloro is another animal that it's nearby, but it's not that one.
Because those metabolites, it's multi-hormone, right? That's from insect. And also some lower plant we found it in these days, right? That is, now it's animal.
Yeah.
It's everywhere.
And it's huge because the first works in Mediterranean, with the species of Mediterranean, the majority of the compounds isolate are with 3 or 4 oxygen, but it's the same scale.
That kind of compounds in before 20, so it's for the student, 20th century, we believe only from insect.
Yeah.
Multi-hormone, the bird, but since then, very low plant, like a Polypodiaceae, they found this plant from plants. Now, we said it's from animal, so which means it's everywhere. So
They are the major compounds. Okay, thanks a lot.
Professor, it's for
Okay, let's thank again Professor Norberto. Metabolic fingerprint by enema. Wait, this. Okay. Just a second. Okay, Professor. Ferreira has a degree in chemistry from the Federal University of São Carlos, exactly here. A master and doctorate in organic chemistry from University of São Paulo, with Professor Gottlieb, both in area of natural products chemistry. He did a postdoctorate at the University of Sheffield, England, in the area of nuclear magnetic resonance. He is a full professor at the University of Federal of São Carlos, exactly here, where he carries out the research in the area of NMR. The main focus of its research is the training of human researchers focused on the area of NMR in the following of the interest. NMR as an analytical tool applied to food analysis, food NMR, quality control of herbal medicine, fingerprinting, and the determination of no specific compounds, no targeted compounds, environment sample. Recently, he received the Aurein Award outstanding in the NMR. I like so much when Gilberto wrote in his CV. The main focus of its research is training of the human resources. It's not common, and exactly we do is exactly he wrote in his. Congratulations for the two cases, please.
Thank you, Fatima, for this very kind introduction. First of all, I would like to say that our facility that we have in our lab came from different groups. And certainly, one of the groups, the most important group, is the ENCT. It is natural products. So many of things that we do is focus on natural products. Of course, something that we do completely different, but the strong support came from these project, but from the natural products. I choose the metabolic fingerprint by NMR, but just as one title, not more than this. On this point of view, I talk about the metabolic fingerprint, compounds identification, and two different cases. Well, I received, my time was less than I have, but so I could and change everything during the lunchtime. So the first thing, fingerprint. Well, we have an explanation, we have a lesson, we have a course about this. Of course, I'm not discussing this, of course, because Professor Choi show clear, Professor Choi show clear the difference of the fingerprint and also the fingerprint is for classification, compounds fingerprint or chemical fingerprint or metabolomics. We're not discussing this because, well, we have a weekly discussion about this. But I would like to point here, like I asked to Norberto, we know that fingerprint, we use typically PCA. Of course, there are some supervisor method, unsupervised method together. But the point here is when you run one of these, what you are doing indeed. Like I said, most of the students in the beginning start to press photo and have some results. But you must to know what is behind this result. Because behind has a lot of mathematics. Normally people say, oh, this is computer. Well, computer is programmation, but who organized the algorithm? So, my advice, because I don't know about the PCA, I don't know, oh, I know the concept, of course, but I don't know how to run a PCA. My student must know, but I don't know. My advice is, when we start to do any measurement that you intend to use one of these tools, it's better if you have in your team someone who really works with PCA or with these tools. And also, when you started to elaborate the problem that you wanted to solve it. Just in the beginning, it's important that you have the guys from the mathematics or, of course, this is not statistics, but it's on the beginning. You have some statistics dependent problem or some chemometrics dependent problem. Start with this guy on your team. Don't just run the mathematics. Don't run the equation because sometimes you have different results. We have some expression here. Chemometrics is the art to compress your results for you get the answer that you want. So, of course, it's not true, but you must take care. The second point is chemical fingerprint. Of course, we can use different measurements, UV, FDR, MS, NMR, etc. And most of the time, or sometime, the student or we see on the literature or in the conference, the people say, well, I identified 200 compounds. I have identified 500 compounds. I have identified near 1,000 compounds. Okay, maybe it's true. Okay, but you must to be careful. What you call identify the compounds. You have to have more evidence that the compound is really inside, if you are talking about the matrix, is inside in that matrix. Why I say this? I say this because most of the time the people don't have strong evidence, and they say, wow, well, not of course alanine, but the alanine is on this matrix. Maybe we don't have the component inside there. So we need to be careful with this, to be careful when you say identify the compounds. So we need to have strong evidence. Okay, the first case is really a simple case. We start print or chemical fingerprint or fingerprint or metabolome. The first point is what the answer that you want, what is the question that you want to answer? What's the question? And in this case, we also have a look about the quantitative NMR. I heard a lot the people say, oh, NMR is a quantitative measurement because the integration of the peaks is proportional for the number of the hydrogen. Yeah, but it's not completely true. Because for you to say this, you must be careful, depending the measurement. If the measurement is not doing in the correct protocol, it's not true. Certainly it's not true. But most of the time, the people say, no, it's a quantitative, just looking for the integration. No, you must take care about this, okay? So the first thing is what we are interested on this simple case is to looking if we can see the difference. Use NMR. Well, why we use NMR? Because I work with NMR on this. You can use other things. If we can make difference between when you put our orange juice, fresh one, on the refrigerator or outside the refrigerator on room temperature, if you can see difference in chemical constitution. Why this? Well, in this country, we normally drink fresh orange juice, normally, okay? The young people now start to drink other things like, not concentrated, but concentrated, diluted orange juice because it's easier. You buy can, buy bottle, but it's easier to find or fresh orange juice. And we know that when you prepare orange juice and you left outside the refrigerator, or depends the time inside the refrigerator, when you drink it after some hours, the taste is bitter. It's not good. Maybe we can see the difference between these two things. The other thing is normally the mothers, the mammies, when introduced for the first time orange juice for the babies, they introduce one specific orange juice, one specific fresh orange juice, not the acid one, for obvious reason. They normally use one kind of orange that we call here lime. So the first experience for the baby orange to drink the orange juice is lima orange. And we suspect, we had suspicion that on the orange juice we found ethanol. So the idea is to solve these three problems. First, it's different inside, outside, depends the time. If you use different orange, we have or not have ethanol inside the juice. We choose to study this kind of three different species: Citrus senensis, Citrus aurantifolia, Citrus limonoides, different species and different variety. Why we choose this variety? We call orange pera, orange Bahia, Morcote, Lima, Lima da Pérsia, grapefruit, and sweet orange. We choose these 1, 2, 3, 4, 5 because this is the orange that normally we found on our local market. So this is the reason. And how we prepare the sample? Well, just squeeze the orange. Of course, we classify by size, by maturity, by species or variety, and squeeze the juice, the fresh one. Okay. We centrifugate to remove the some particles, of course, add buffer and sodium azide to stop the enzymatic procedure, and we prepare one stock solution with D2O and TMSP4, TMSPD4 to decrease the error. So this is the way that we prepare the sample, and just add the orange juice. So, like I said here, we just prepare a solution stock, and we put the same amount of the juice inside and outside the refrigerator. The temperature 14 and 24, 25 is because the temperature of the lab is 24, and we can control the temperature, and inside the refrigerator is 14, because inside the refrigerator is 14, and we keep these two temperatures under control. And of course, we measure the NMR measurement. The first 1 hour, 2 hours, 3 hours until 12 hours. The police, 24 hours, and the 1 day, not more than 2 days. Okay. And of course, we use here this detail of experiment. And you see now, in that time, you use one kind of software to produce the PCA. Unscrubble, but it's one of them. Mix is another one. There are a lot of software. This is the spectral one. We can identify these compounds, and what I call identify here. We run here the 1D, of course, the hydrogen, and also the 2D spectra, COSY, HSPC, HMBC, HSPC, toxic, sometimes chain modulation, to get evidence that really here is arginine. It's not only because the software say, oh, this is arginine, because we have more than one evidence. And of course, this is the region of the aromatic region, and there are something important here because fluorine is some history about fluorine. Fluorine Normally we find the fluorine in the right peel of the orange. If you remove the peel, you have the right peel, and fluorine we find normally on this right one, albedo. I don't know, albed in English. And this is one compound that, if you look here, say, I would say this is fluorine. You run a 2D experiment, forget it. You cannot see, you cannot identify fluorine here. But when indeed these results come from one company, they looking that this peak is very important. They isolate the compound and identify the compound. It's fluorine. And this is one chemical, this is one compound that is a biomarker for the Brazilian style to produce orange, concentrate orange. Because it's not allowed, but it's possible to wash the albed to remove a little bit more juice, but you also extract it. Not necessarily extract, you remove it more fluorine. So fluorine means this coming from Brazil, juice from Brazil, or from the people who don't take care about it, not to wash the albed. So, what do we have here? Well, it's a simple PCA. There are two things important here. First of all, this orange that we call, well, I say N1, N3, N2 is it's easier, but you can read it here in Portuguese. So N1, N3, N2, you see, don't make so many difference if you store on the refrigerator or not. It's the same, or near the same. But we know that the taste doesn't matter if it's N1, N2, N3, N2, the taste became bitter. In another word, we cannot discriminate by the taste, only by the taste. But if you looking for the N4 and N5, what you observe, there are a big difference when the orange juice is stored inside and outside and during the time. If you look in here, 24 hours different, 24 hours is different. If it's inside or if it's outside. The same here. So we can discriminate this. This is fingerprint. We can discriminate. We can see this is one kind of orange juice, is another kind of orange. But of course, the second point is, what is the compounds responsible for this? So we need to know what is the compounds. And the compounds that you observe that is responsible, especially for this kind, Lima, N5, and N4, don't forget, this is the orange that the first time the man introduced for the babies. So we are looking, we have for all these orange juice, but we are looking for the orange juice. When we're looking for the orange juice, you see the succinic acid, acetic acid, and lactic acid increasing substantially when it's outside the refrigerator and 24 hours outside the refrigerator. So these acids probably is responsible for the bitter taste. And also you observe, of course, the sugar, the content of sugar decrease. Of course, they decrease because the enzymatic process convert the sugar in this aspect. But we have another information that's very important. Is the ethanol content. All the juice inside the refrigerator, outside the refrigerator, for a short period, for a long period, have ethanol content. Depends, is more quantity of ethanol, or like this one, after 24 hours, inside or outside, outside or inside the refrigerator, the content of the ethanol increased substantially. And if you look here, this is the orange that the man introduced for the first time for the babies. In another word, if you prepare, you're a lazy man, I suppose no man is lazy, but if you are a lazy man, what do? You intended to produce a good food for your baby, and you prepare in advance because during the night you cannot prepare or you don't want to prepare, and if you stay inside or outside the refrigerator, it's a normal one. You can put it on the baby bed, just became easy, but the content of ethanol increased substantially. So indeed, you are doing more ethanol than alanine. Alanine is essential, of course, for the baby. We compare the ethanol content. I do not show you here the spectra, but we observe this. The ethanol content on this situation is 10 times more than the alanine content. So you give alcohol for your baby. Okay. Of course, you can use this excuse if you go to the bar and became in the house drunk. You say for your boyfriend, girlfriend, or for your mother, or for everything that you drink a lot of oranges. You can use this. I tried one time, but my wife did not became happy with my excuse. I don't know why, but this is some results that we had. So it's a simple question that we try to solve it using NMR. And in the simple way, just prepare the sample and add D2O with ESP, etc. in the protocol that's we founded. We follow the protocol for all the measurements, of course. But it's a simple way. Just mixture, orange juice, and water, deturated water. Of course, we do give all these fruits for in the end you understand why we study fruits or juice indeed, and other things like honey, coffee, soybean, vinegar, cachaça, and açaí. I explain about açaí, and also for some vegetable that normally is used in the medicinal way, especially in Brazil. Well, there are ginseng here, or this is not ginseng, of course. It's a Brazilian ginseng. It's not have any correlation with ginseng, but the people use. And why they use? They use only because the root is similar to the true ginseng. Don't make a sense. But the people do this, okay? So also these two species here, this species is completely, the family is completely different. So one is used for one purpose, it's described in the Brazilian pharmacopoeia, the other one don't make any sense. But the people make some confusion. If you go to the supermarket or the market, you can buy the people, can ask the people, and you can buy both of them. Now I would like to talk about acai, but not only about the fruit, but about to use the enema with another purpose, to found small compounds, the minority compounds, the compounds that is in the minority inside the matrix. Okay, everybody knows acai. This is the fruits, this is the pulp, and we found this acai on the supermarket. Normally not here in the south, but most in the north. One thing that is very important with the acai, acai degrades very quick. So we have to, we need to have a strict protocol to collect the sample. On this case, we went to the north, on the place who is specialized in acai and brapa, and we looking for what is the protocol that they collect the acai. And it's very important to collect in the morning, very quickly. Remove the coconuts, small coconuts. There are only the small peels. It's these small peels that they produce the pulp. But they remove this, weigh this, count each one, and produce the pulp immediately. And after this, put on the refrigerator -20, and when it's a solid one, lyophilize this. Because otherwise, the compounds, they start to decompose, and you cannot make any useful measurement. So, what was the procedure? Is as I described, and as they use it, normally use, they try the pulp, lyophilize the pulp, and store in the powder form. Okay, so we use the powder one, lyophilized powder, and well, okay, this is something very easy. We weight the powder, the same weight the powder, take a lot of care about this. Of course, filtrate solvent directly. In this case, was methanol with DSP-T4, not TNS, because for obvious reason. Normally, we use, like Professor Choi showed, the TMS or TSP-T4. It's used for correct the spectra, centralize the spectra, but also to check the sheen quality. And we submit to ultrasonic. Here we observe that if you use one batch and submit to ultrasonic, after 15 minutes, the second batch changes the temperature. So we put it, the same batch, the same temperature, and all the batch there. So centrifugate and filtration in a filter with this size. And this is something that is important. for in my opinion. What we did, different extract, methanol extract and acetate extract, and made some chromatogram. We do not use open column. Of course, everybody can imagine you use this extract in an open column. When you finish, you don't have nothing useful. So we run HPLC, and what we did, we chose one of these peaks because we had a lot of compounds here, of course. One of these peaks, and after this, we optimize the HPLC condition only for that fraction, the fraction that comes from one of these peaks. It's a semi-preparative column. And one of these fraction, and this is the HPLC. If you look in here, normally when you look in a chromatogram, normally we make 3 mistakes. It's typical, 3 mistakes, but it's good to think. It's good to make a mistake. The first one, this is a very, well, not very, but it's a thin bit. And probably it's here, pure compound. And not necessary. It's not because the peak is separate that the compound is pure. On my experience, when you take some fraction from HPLC, never, forever, it's pure compound. When you use a matrix, of course. The third mistake is, well, I have the peaks 1 to 8 is the most abundant compounds present on the matrix. Maybe it's true, but maybe not. Depends, because I use ultraviolet, indeed, the DAD, but doesn't matter. It's ultraviolet. If we do not have a good chromophore, the peak is more intense because I use the ultraviolet. And the third error, or the third mistake, maybe, say, looking for this. There's nothing here. Well, maybe it's true. There's nothing there. But not necessary. Maybe there are some compound in small quantity and not good chromophore. So we cannot see on using ultraviolet. So, of course, we analyze the big one, the big peaks, but also the small one. All of these, of course, my student was not happy with this. In the beginning, he was happy because they say, well, it's very easy, identify everything, but are you sure that you wanted to trap this one and try to analyze? And what's the way that we trap this? We use solid phase extraction. We started to use NC NMR, direct. Stop flow and on flow. But we don't have success enough. NC NMR on flow, definition, doesn't work well. So the best way is to concentrate the sample using SPE. So, oh sorry. Very position, but of course we concentrate the peak. After this, we dissolve the peak in methanol and run all into the experiment, NMR experiment. But if you look here on this region, we can see nothing. So I asked for my students, say, you take some sample from here. There are different ways that you can take the sample. You can use threshold. Threshold is very nice because it's very nice when you have a big, intensive peak, you select it by threshold and say, well, on this threshold, you can select. But for a small one, doesn't work well because it's near the background noise. The other one is time sliced. So you define from this time to this time. Normally, when you have a very intense peak, we use threshold. When we have a small peak or a small intense peak, we use time slice. But if you're looking for this situation here, we cannot see nothing. So if you trap this region by time slice, they say, oh, from here to here, you trap this, you put in the solid phase extraction. And if you don't see nothing, the student will say for me, I told you, there is nothing here. This is the reason I waste my time. But we have another spare part or another part of the instrument that Brucker called PPSU. It's a collect. It's a loop storage. It's a fraction collector. So what I ask for the student, collect the fraction and dry the collected on this tube. Dry the sample, and after this we have a look if we have in the sign. Because if I have sign looking for the point of view of the animal or the point of view of mass, we have a compound. If you trap and don't have compounds, it's because the SPE cartridge is a wrong phase. We need to change the phase. Fortunately, of course, we trap this and we observe the spectra. The spectra from the trap of this region using the PPSU. Of course, you have a compound here, but this compound is not the majority. The compound here is not the majority peaks here. Of course, it's a small one. So we need to run more time machine, and we identify the compound here. The other one is the compound that is contaminated on this region. So we identify this compound. Like we suppose we have compound here, but the chromophore is not a good chromophore, so don't have a good intensity, and also we have a small quantity, really small quantity. So we identify many other compounds, 23 compounds, using the same approach. So using the LC-UV-SPE, in my opinion, this is the best combination to concentrate compounds. But of course, you have a lot of job here. First, you need to develop the chromatographic condition. Otherwise, if you don't have a good chromatographic condition, you cannot concentrate the compound that really is useful for the identification. Most of the time you have a massive mixture. And of course, this unit, it's important if you are looking for compound that it, you know, they say, well, I do not expect that compound is here. Maybe you can use this this unit. Well, what is the future? We also work with metabolomics, but with the body fluids. We have some examples with body fluids, but I will not show today. But in the future, for the people that we are listening to us, we both, well, we expect that we receive one IVDR protocol, one IVDR platform. What is this? For the people, it's outside São Carlos. It's a 600 megahertz with BBI broadhead. It doesn't matter the details. With a lot of spare parts, or sorry, a lot of different combination of equipments. Sample eject. This is a controlled temperature sample ejected. We have also sample preparation, automatic sample preparation to use for metabolomics, not only for body fluid metabolomics. Of course, the IVDR platform that includes all these equipments, but also the software, the algorithm to analyze body fluid, especially urine, serum, and blood, but also to analyze extract from vegetable. The IVDR is not specific for vegetal extract, but our idea is to use all these systems for only metabolomics. This is an equipment that we will have probably in the next year, in the middle of the year, and it's a multi-user equipment, and also we try to use it as facility. In other words, the people who is outside of São Carlos can, if he had a training and if you know how to use, the people can come here to São Carlos or remotely can access the equipment and make the measurements. So this is that we think the future for the lab, the near future. Also, I would like to invite all of you. This is the reason because I studied food enema, because food enema is an ampere. Food enema is part of the ampere. It's around the next will be the 16th. It's a big annual conference. So it's 32 years old, but incredible. The people in Brazil do not know this one. And this is the first time that MRI food will be in Brazil, of course, but outside of Europe. This is an event typically from Europe. For instance, Wageningen, we have one time in Wageningen. And also, this event will be together with the Brazilian meeting, both in Foz do Iguaçu, in Paraná, and in this data. So save this data. This is the reason because I like to study food or food enema. The other reason, because if you choose the correct food, but the really correct food If you are not happy with the results, you can drink or eat the food and maybe special beer, special cachaça. You can drink the matrix, you can drink the sample. You can find more information here. I will be one of the chair of this event in July. So I'd like to say thank you for all these financial agents. Like someone of you knows, I don't know if all of you knows, but certainly the people outside of São Carlos, the people is virtual way, don't know that our lab has GLP condition. We work on this GLP condition, PPL, and this is the label from the Inmetro. So we are recognized by official agents that we work on the GLP condition. And of course, thank you for your attention.
Next time for question. Okay.
But I worked with, I studied the orange juice in the same way, producing like as you produce in your house and keeping in laboratory during the day, and I got 22 degrees, and kept it, and we quantify vitamin C each 30 minutes during 6 hours. And we checked during this period, the concentration of vitamin C reduced 30%. So why you not detected vitamin C in your studies? Maybe you check what is the pH of your final solution that you study?
Well, I don't remember exactly the pH, okay? We put a buffer, okay? But I don't know exactly the pH. I don't remember because this study was 10 years ago, so I don't remember. But there are two reasons. One reason because the pH is different.
Because vitamin C is not stable in a pH above 4.
Yeah. So I can't remember what's the pH, but this is one reason because it's not stable. Yeah, problem. I think this.
Maybe the sodium aside became the medium base.
Yeah, maybe.
Because on pH 7, you almost doesn't have more vitamins.
I think that is not pH 7. I guess it's acidic. When I look at his condition of pH, I think it sounds like acidic condition. Because also, even he did not mention pH. When I look at the citric acid and malic acid, the signal, it looks like acidic condition.
Yeah, but I don't know if it's enough because, for example, the condition that I use working real. My solution of the mobile phase was 1.5. It's only water with sulfuric acid.
Also. May I look at the animal spectrum? Maybe I can give us some the animal identification in orange juice. You identify the compounds? Yeah. I think it's if there is ascorbic acid, vitamin C, ascorbic acid,
Yeah.
it's overlapped with the 4.0 ppm. So we don't know whether he could detect or not. I mean, this one he can
it cannot distinguish, which means it doesn't matter for his research. But you are right, if you want to target the vitamin C, ascorbic acid, around the 5 ppm, very small triplet, you have to detect it. But in this case, there are a lot of other phenomena.
And another question about the bitter taste of orange juice. The literature described that the main reason is because limonene is converted to limonoic acid when you store the orange juice. And carnitine and aspartame, when hydrolyzed, they contribute to a little bit of this bitter taste.
Yeah.
You, you do not observe
Or not.
limonene and
Yeah, not on this condition, because the quantity, but you are correct. Maybe we have some small compound of the compound with small amount that is responsible or more responsible for the bitter taste. But we just identified that acid is okay. So well, limonene maybe it's the major response.
Yes.
But we cannot see on this condition.
No, no.
that's when limonene is bitter.
It's bitter.
Yeah.
And the limon
But-
is extremely.
No. But in this citrus, the bitter taste usually is not from limonene, lemon, lime, probably from limonene. This case is naringeline. Usually flavanone is but
Yeah.
actually that was my question, but I can continue after you. I will continue. But it's not limonene. I don't think maybe there is, but if any, very small amount in
Yeah.
orange. But in lemon, lime, green and yellow one, it should be lemonade. Triterpenoid.
Yeah, anyway, we cannot detect.
No.
I have a question, a similar question. Then you have
Yeah,
a
sure.
question.
No,
Last week, I analyzed one paper on they just changed the graft so that they can have orange, sweet orange resistant against a green bacteria. And they analyzed the juice exactly because can you imagine the graft changed the juice? The juice became more bitter. And then they analyzed and quantified. And the limonite, limonene, and the same flavonoid you said.
Yeah, but in this case,
Yeah.
you must to use the MIS. Probably you use the MIS in the art study. No?
No. The student I said it's from literature. I received to analyze last week.
Yeah,
I can
but
show you.
he studied using which technique?
PDA, quantify the vitamin C
Okay.
by PDA
Okay.
and the
Yeah.
conversion of limonene to IKF.
Yeah, for point of view, the resonance is kind of effective.
And I'm very curious how you can have ethanol. Because if you have acetic acid, it's very common when you have a microorganism, you have an oxidation with no reduction. But of course, it's possible that you have a bacteria or fungal and you can have reduction.
Yeah.
But I'm very curious. So
It's from microorganism.
yeah, yes, I think so. Yeah.
Yeah, it's not. Yeah, you see, like I told you, the ethanol content and alanine. And another thing curious, not me, but Conago, who works also with fruits or juice, he found the ethanol in the coconuts, inside the coconuts.
Yeah.
It's very common. It's in,
Yeah.
in plants, mate, whatever, you can detect ethanol, very common. And next one, ethanol, next one, if I could advise, this one is ethanol glycoside, ethanol glucose.
Ethanol glucose.
Really?
I know that. But,
Really?
and may I continue this one?
Yeah, of course.
In this kind of presentation, it's not very good. It's not very good etiquette but may I have some doubt about the identification?
Yeah.
Because succinic acid, you said, is a 2.5 ppm triplet. Succinic acid, 2.5 ppm triplet. Yeah. But you showed next slide, succinic acid is very changed by.
Okay.
By,
Yeah.
Actually, succinic acid should be singlet. It's a triplet means. I guess this one is rather. It sounds like a GABA, which are produced by microorganisms. Then your story is correct because after keeping high temperature, GABA increase because of microbes or not. I guess it sounds like the GABA signal in any case. And fumaric is the left signal, the left of fumaric acid. You didn't identify big signal, the left. No, the other side. No, in between.
Between both.
6.7 ppm. Yeah. No, left. Yeah, that's one. That one is a ischemic acid glycoside. And the big signal tyrosine and the 5-hydroxy methylpurfural, that case is those big signal. I guess this one is the flavotannin cases. So those around 6.8 ppm and 7.6 ppm. You have two big signal. Always that kind of signals when you get from food, it looks like a bitter taste, tannin case, flavotannin cases, I think.
I think so.
But animal identification is always also based on assumption, assumption by.
But there are some straight attribution mistake.
Yeah. But then also this one is sometimes is from plant. When you search the like a Genotype-Environment, the mammalian database, human metabolic database, many people misassign. That is a problem. But this succinic acid, I think you can have a look the singular the identification. I'm sorry, sir.
It's okay. Yeah.
This is what it is.
More question? Okay, let's again. Thank you, Professor. Okay, I have some words before I close this session. Okay? For everybody, dear colleagues, you have been here for 5 days, and I hope you enjoy the event. At this moment, I would like to extend, on behalf of the organizing committee, our thanks to Professor Hae Choi for the excellent course. To all the lectures, I'm happy to announce that the event has attracted over 261 virtual participants or in the YouTube channel by SPOK and the Division of Natural Products. Again, we would especially like to thank the Natural Products Division of the Brazilian Chemical Society. Then I say thanks a lot for Felipe and all the other colleagues from the same division. To the students who became involved in the organizing of this event. Thanks a lot. To Professor Antônio Gilberto Ferreira by intense dedication in the activities during the several preparative phases of this event. Thanks a lot to Gilberto. We are also very grateful to our colleagues and the students for what they have done to make this meeting the success, which I think it was a success. We declare the event closed and thank you all for your attention. just all right.
Can I say something?
Yeah, of course. Professor Fatma say thank you for many people, but indeed we must to say thank you for Professor Fatma.
Okay, now we can discuss